My name's Elaine Stokes. I'm an account manager and facilitator for BioPhorum. It's a very great pleasure to introduce this bioassay webinar, which is the first for the development group. I'd like to introduce the speakers who are from around the globe in the order that they will present. So Jeff Talbot is group leader from Analytical Sciences at Regeneron, New York. Samina Ahmed is senior group leader and principal scientist in the biopotency and immunoassay group at Lonza Biologics in the United Kingdom. Gail de Bouw is head of gene therapy and CMC analytics at UCB in Belgium. And it was Gail that first proposed the idea of a BioPhorum bioassay workstream way back in 2015. And finally, John White, who's Director of Biological Methods in CMC Analytical Group at GSK in Collegeville, Pennsylvania. And John proposed the idea of drafting our best practice paper. So we're going to start our webinar today addressing mechanism of action considerations, and then we'll get into the tactical design of bioassays. We'll move on to cover the all-important analysis of data and system and sample suitability, followed by approaches to monitoring bioassay performance. And then we'll round off the presentation with conclusions and future perspectives. Can I have the next slide, please, Nettie? BioPhorum consists of 7 forums, and it exists to create an environment where the global biopharmaceutical industry can collaborate to accelerate their progress for the benefit of all. The development group's mission focuses on better and faster process development of biopharmaceuticals. We bring collaboration to the drug development community while navigating the challenges of being close to intellectual property. This webinar is presented by members of the bioassay workstream, which is one of the 13 workstreams within the development group. So if we go to the next slide, this tells you a bit about that bioassay workstream. Since 2015, blinded surveys have been drafted and completed by the workstream participants, and we've had a very lively discussion on the monthly calls, and we didn't always gain consensus for best practice. And in these circumstances, we share all of the approaches used. We've documented the output of these discussions in a publication which came out in Biotechniques last year. This presentation is a composite view of opinions shared by the whole of the bioassay workstream and should not be attributed to the individual positions of the participating companies. So today we will present best practice in bioassay development to support the registration of biopharmaceuticals. And on the next slide, you will see those authors that have drafted this publication and their hard work to deliver the paper. I'd also like to acknowledge the key members of the bioassay workstream, past and present, for their survey results and discussions, and they're also listed on the slide. So now I'd like to go to the next slide. And Jeff will be our second presenter.
Thanks, Elaine. Before I begin, I want everybody to know that the majority of the following discussion will reflect recommended approaches for recombinant antibody therapeutics. This section of slides focuses on the importance of understanding a candidate's molecule's mechanism of action and the impact on assay design. Subsequent presenters will go into greater detail on tactical components of bioassay design. That said, let's begin. The mechanism of action is the force that drives and focuses the development strategy conversation. A lack of understanding of mechanism of action can lead to poor assay design and an ineffective development strategy. Before we move on, I'd like to summarize this section of the slides in advance. The mechanism of action really is the tail that wags the bioassay development dog. Next slide, please. So why is understanding the MOA important to assay development? First, global health agencies all require a potency assay. This is because the potency assay is the only analytical method which quantitates the candidate's MOA, potency, and associated higher order structure. Because of this unique MOA reflectiveness, each candidate typically requires a unique, custom-built, fit-for-purpose assay. Because of this, bioassay scientists almost always have to start the assay development process from scratch each time a new candidate enters the pipeline. That said, since MOA understanding typically develops as the program progresses, it seems daunting to develop a deeply MOA reflective assay with incomplete knowledge of the mechanism of action. Agencies are aware of this challenge and generally are in agreement that a phased approach to assay development should be taken. This means that for candidates with simple MOAs, a binding ELISA or SPR style assay will typically be acceptable to carry the program into the clinic, with the expectation that a more complex, physiologically relevant assay be developed for pivotal trials. If additional MOAs are discovered during product development, the expectation is that those MOAs also be captured with a potency assay. Whether they are on the release or the characterization panel would be a conversation to be had directly with the agency itself. Next slide, please. Here we have some cartoon examples of simple MOAs. These would include antagonistic activities, such as binding to a cell surface receptor or soluble ligand to prevent binding and pathway activation. Agonistic activities, such as binding and cross-linking of cell surface receptor subunits for pathway activation. In terms of capturing simple MOAs in an assay, ICH Q6B is supportive of the use of binding assays, particularly for mAbs, since in vivo, the mAb must first bind to induce its biological effect. Historically, the cell-based assay has been the bioassay gold standard for pivotal trials. However, a survey of member companies found increasing frequency of discussions with regulators regarding the use of binding assays for pivotal trials. At the end of the day, the decisions should be driven by the candidate's MOA, so an in-depth understanding of the MOA will help in developing the data set to support this approach. Next slide, please. Here we have cartoons of complex or multiple mechanisms of action. These might include ADCC activity, CDC, or ADCP activity. Typically, these MOAs include multiple cell types, multi-specific candidates, and more than one biological effect being induced. Sometimes these secondary MOAs are not intentional parts of the design process, but rather are discovered as product knowledge grows. One of the main challenges with these multiple MOAs is capturing them all with assays and deciding if they should live on the release or characterization panel. Next slide, please. With an understanding of the candidate's MOA, that lets us then start thinking about how to capture it appropriately in an innovative, modern bioassay design. Technology advances have moved us away from the legacy multi-day proliferation assays with colorimetric readouts towards a single-day reported gene assay with a luciferase readout. With these modern assay formats, the advantages typically include ease of use, assay length, sensitivity, and dynamic range. These benefits also allow for higher throughput, a lower failure rate, increased ease of transfer, and overall lower assay cost. As you can see diagrammed here, the luciferase reporter gene and secondary messenger assays have been found to be in wide use across member companies and are well received by regulators. Interestingly, new non-cell-based technologies have emerged, including homogeneous proximity-based readout assays. However, a survey of member companies found that this format has not been widely adopted as of yet. Next slide, please.
Okay, thank you, Jeff. In the tactile design section, we will discuss the components of a robust assay, beginning with selection of cell line, moving through to assay design by plate layout and identification of bias, plate effects, plate randomization, and replication strategy. There are several inputs that are needed to establish the final potency assay format prior to qualification, and qualification describes activities which demonstrate the suitability of a method for use in early stages of product development, whilst a full method validation is defined by using a pre-approved protocol with acceptance criteria and is performed prior to using a method for later stages of development and commercialization. Next slide, please. Next slide, please. Thank you. As highlighted by Jeff, cell-based assays are complex and variable. And to control for this variability, a A methodical stepwise approach is used to design and select the assay type, which best reflects the MOA and subsequently has a direct impact on cell line selection. The choice of cell type is between a primary cell line isolated from human blood or tissue and an immortalized cell line from human or non-human origin. Modified cell lines can also be considered, which overexpress the receptor or ligand that is targeted by the therapeutic. Where the MOA of the therapeutic agent is complex or multiple receptors are involved in the MOA and a transfected cell line cannot fully represent all the interactions, a primary cell line is the default option. There are several considerations for selecting the most suitable cell line, which involves the availability of numerous cell lines per cell type, several cell sources, and identity testing. Donor-to-donor variability from primary cells can be partially circumvented by freezing of banks and utilized in a ready-to-use format in the bioassay. Whether the ultimate decision is to use cells which are prepared for a ready-to-use format or continuous culture, a strategy to ensure reproducibility of cell banks is proposed, and this includes assessment and optimization of passaging frequency, cell seeding density, cell doubling time, monitoring cell viability and receptor expression, selection of FBS type and quality to prepare highly consistent cell banks and ultimately will lead to a consistent bioassay performance. Next slide, please. We'll now review microplate assay variability. The 96-well microplate format is typically amenable to most binding and cell-based assays, and this format allows the use of commonly used equipment in most laboratories, multi-channel pipettes to prepare and add dilutions, reagents, using plate washers and plate readers that facilitate the performance of these assays. Plates with higher well count can also be considered, but would require an investment in automation for routine use. It is recommended that both manual and automated methods should be assessed using the same reagents in order to allow flexibility as to which assay is implemented in a QC environment. There are multiple sources of potential variability in using microplate assays that can affect assay performance and thus the accuracy of results. And these include uniform cell plating or protein coating, inconsistent growth rate and or response, biased results due to the location of the sample on the plate, order of standard controls, samples and reagents addition, and variability from analyst to analyst, plate to plate, and run to run. A number of approaches have been reported in literature and discussed by Bioassay BioPhorum members to minimize or protect against potential plate location effects. And these include inclusion of replicates, be they wells or plates, a strategic consideration for the use of a standard control and test sample position on the plate, randomization of plate layout, exclusion of outer rows or columns, and automation. Next slide, please. The most common plate-related phenomenon is the so-called edge effect, where the response from the peripheral well differs from the inner wells of a microtiter plate. And there are two approaches which may be used to evaluate this effect. This includes using a full dose response curve using the reference standard in the final plate format, with the ED50 values averaged for all plates and compared across multiple plates, or as shown in this slide example, a single concentration targeting the ED50 value is plated in all wells and the responses compared from the exterior wells to the interior wells and graphed to determine the extent of bias. If no edge effect is observed, all wells can be used. However, if an edge effect is observed, then the common mitigation is to restrict the usage of the plate to the inner 80 or 60 wells. One obvious side effect from this approach is reduction of sample throughput, and to avoid reduction of sample throughput per plate, alternative approaches to reduce the edge effect are recommended to be considered. And these are use of circumferential built-in moat microtiter plates, plate hotels, heat transfer plates, or pre-incubation of cell seeder plates at room temperature prior to initiating the assay. Next slide, please. A second approach to minimize or protect against plate location and plate-to-plate bias is pseudo-randomization. This can be applied within a single plate or between plates. Within the plate, dose replicates can be loaded in alternating rows or columns, and plate location effects can be mitigated by ensuring all samples are equally exposed to the edges. The choice of a number of replicates may vary based on the application, final application of the result, whether it be for release, stability, comparability, or characterization, and also variability of the bioassay. Commonly, at least 2 replicates per dose level of reference standard control and samples are used within a single plate. And when needed, 2 or more plates are used to generate the sample reportable value. This replication strategy should be established through statistical analysis in order to achieve the desired method performance to support the specification. And here we do advocate consultation with a biostatistician is very beneficial to support the long-term bioassay strategy. Next slide, please.
Thanks, Amina. It's now time to move to the way the relative potency results are analyzed. I think that the first consequence of a relative potency analysis is the fact that we have to consider 2 dose-response relationships. One for the unknown sample, that is called the test sample preparation, and one from the standard. The second specificity of biological assay is that the dose response usually present a non-linear relationship, and this is a big shift compared to physical methods. So that non-linear relationship is usually characterized by a S shape. If we go to the next slide, we see a graph that represents the dose-response relationship between a standard and a test sample preparation. On the x-axis, you have the representation of the log concentration, and on the y-axis, you have the assay response that could be translated into fluorescence unit or into absorbance. So the relative potency is here represented by an horizontal shift between the 2 curves. From a mathematical point of view, you can compute it as the ratio of ED50. And ED50 is the concentration point where you have half of the effect. So it's really the midpoint between the upper and the lower asymptote. The recommendation of USP is to consider the full curve model compared to only considering the linear part. If you go to the next slide, in that slide, we define the key concept in analyzing a relative potency value as part of the similarity assumption. So in other words, the relative potency value would be meaningless if the 2 compared dose response relationship does not share the same parameters. So by same parameter, I mean the asymptotes and the slope. Indeed, as relative potency is the reflect of the ratio of ED50, ED50 should be influenced by the horizontal displacement only. So in the following graphs, you can see examples where ED50 could be influenced by slope differences, so on the right-hand side, or by asymptote differences, so at the bottom of the slide. Consequently, there must be a way to measure and to test the curve similarity or parallelism as a prerequisite to calculate the relative potency. And to that specific question, 100% of the BioPhorum survey responded confirmed that they do test similarity as part of the sample suitability criteria. So if we go to the next slide, we here propose a 3-step process to define the similarity assessment. So the first step resides in the selection of the most appropriate fitting model. And the model that is commonly used of curve fitting analysis of symmetric sigmoidal curve is the 4PL regression model. So the 4 parameters of that model consist in the upper and the lower asymptotes that are traditionally represented by the A and D denomination. You have B that represents the yield slope and the C that corresponds to the ED50. A sigmoid function requires a sufficient number of concentration points to fit the model. And the BioPhorum bioassay members recommend a minimum of 8 points. So 2 points to characterize each asymptote and a minimum of 3 and ideally 4 points in the linear part of the curve. So whilst the 4PL is typically employed to fit bioassay dose response relationship, when biological mechanism of action produces a curve that is not symmetric around the midpoint, alternative fitting model might be of interest. And I'm especially thinking about the 5-parameter fit model. Anyway, the validity of the mathematical model should be assessed. And to this end, some approaches are proposed in the literature, and you can find easily R-square or sum of square errors. Again, plotting the residual against the model would allow to evaluate if the model is appropriate. And this is a random distribution or not, that is a systematic deviation from the model. It has to be noted also that weighting approaches can be considered to address fitting issues. So this is the first step. If we go to the next slide, you will see the second step of the process. So once you have the right fitting model, then you need to find a way to measure similarity. And again, you have two approaches that are proposed in the USP 1032 to evaluate that similarity. The first approach is to look at the curve parameter ratio. And the main advantage of that approach is that it is easy to implement and to interpret. The drawback of that approach is that it requires to define the limit on each ratio. The curve parameter ratio is the approach that is the most used by the BioPhorum survey respondents. You can see here 63% of the BioPhorum survey respondents are using that approach. And by curve parameter ratio, you can vary different curve ratio. You can analyze the yield slope ratio, so the ratio of the B parameter. You can also assess the lower and upper asymptote ratio independently, but you can also combine the ratio between the lower to the upper asymptote. So this is the first approach. The second approach that is less popular because it is only used by 19% of the BioPhorum survey respondents is to consider only one measure that is called the single composite measure. The main advantage of looking only at one single composite measure is that you only have to define one limit on that measure. The drawback of that approach is that it's more difficult to implement and to interpret. One example of single composite measure is the residual sum of square approach. That measures the difference between the residual variability of a constraint model, where you force the slope and the asymptote to be equal, and the unconstrained model. And basically, the residual sum of square error corresponds to the energy you have to put in order to force the two curves to be similar. If you go to the next slide, this is the third step of the process. So once you have defined a good fitting model, once you have defined an appropriate measure to look at the similarity, it's interesting to define acceptance criteria on that measure. The main goal of these acceptance limits are to being able to discriminate between parallel or similar and non-similar dose response curves. The recommendation is to use equivalence test because they present the advantage not to penalize results that are too precise. That is the drawback of classical hypothesis test. The problem with equivalence test is that you need to generate historical data. And the size of the dataset will have a direct influence on the equivalence limit reliability. So the general recommendation would be to have a phase-based approach with equivalence limits that are reassessed periodically. If you go to the next slide, that slide illustrates that once the similarity is demonstrated, you can compute the relative potency as the ratio of ED50. And here you have two possibilities. Either using the ED50 ratio from the unconstrained model, so the untransformed data, or using the ED50 ratio calculated from the model where the slope and asymptotes have been constrained to be similar. There is a huge debate, as the classical approach is to calculate the relative potency from the constraint model. However, the BioPhorum survey respondent recommend analyzing data, for example, the qualification data generated using both models, and to choose the ones that exhibit the best performance. And by best performance, I mean the lower variability and bias. If you go to the next slide, we will talk a little bit about system suitability consideration. So before calculating the relative potency, an assessment of the assay performance should be conducted to ensure the assay was in control state during the testing. This suitability assessment can be performed at 3 levels. If you go to the next slide, you can evaluate the system suitability at the level of the system. characterize the quality of the reference standard, to control the quality of the dose-response relationship, the proper functioning of the bioassay system. For example, by looking at the signal-to-noise ratio and looking at the variance in the bioassay run. The second level is to look at the sample suitability. And the sample suitability criteria will compare the performance of an unknown sample to the performance of the reference standard. For example, it might be interesting to look at the goodness of fit, at the similarity, and the replicate variability. And the third level, that is an intermediate between the system and the sample suitability, is to look at the control suitability criteria, where you look at the performance of a known sample compared to the reference standard. What is important to note is that these criteria are evolving in parallel to the method. So they are established during assay development, confirmed during validation, and they may be revised based on performance monitoring or when a bigger dataset is available. And typically, this is done later in clinical stages. If you go to the next slide, and this is also part of the data analysis, you might be facing two abnormal data points with a tricky question on how to address these data points. Some methodologies are described in the USP 1032 regarding the outlier, but the key message is that visual outlier inspection must be supplemented by an objective, statistically derived approach that could be based on an outlier removal tool like the group test, or based on ranges defined on historical data. Outlier analysis tools are used to varying degrees by the forum bioassay member. There is an almost 50-50 split between the companies that have implemented an outlier program versus the ones that does not have any outlier program in place. However, amongst these companies that do not have any outlier program, they have it in the pipe, so they are planning to implement such program in the future. So the main question or challenge associated to outlier removal is that it can be viewed as picking your data. To avoid that situation, you have to put in place clear rejection rules, such as defining how many points you can throw out before the assay becomes invalid, to mitigate the impact of outlier removal on assay performance, and also the impact on the uncertainty on the reportable value. If you go to the next slide.
Okay, thank you, Gil. We'll continue now looking at considerations for monitoring bioassay performance long term. Next slide, please. So following on from Gail's review of sample and suitability considerations, the use of controls are crucial to ensure consistency in method execution and performance. And these include the use of general controls to ensure that the method was executed correctly and performing as intended, but perhaps more relevant in a bioassay format is the use of an interassay control with specific acceptance criteria which serves to evaluate if the bioassay is technically valid and if the potency results should be reported. A review within the Bioassay BioPhorum members has assessed how this is executed in terms of control sample material, secondly, acceptance criteria, and thirdly, timing of implementation. And I'll discuss these individually. Next slide, please. So the common characteristics of an interassay control are that the material should be of a known activity. It should be an independent batch of drug substance or drug product or reference standard. It should demonstrate a comparable level of accuracy and precision to the reference standard. And the results from a survey performed within the Bioassay BioPhorum group showed that 55% of respondents transitioned from using the reference standard independently as prepared as an interassay control or IAC in early phase to a different lot in later phase development, with 18% using a different material lot as an interassay control throughout phases and 18% using the reference standard as an interassay control throughout all phases, with 9% changing the IC at the time of commercial manufacture, aligned with the reference standard change. But in agreement, all the bioassay members did agree the inclusion of an interassay control as a component of your bioassay and long-term bioassay monitoring. Next slide, please. We'll now move on to discussing acceptance criteria. Next slide. Thank you. The purpose and application of an interassay control should be as follows. It should be tested as a full dose response curve. The dilution format should be similar or identical with the sample and reference standard. Thirdly, it should evaluate if the bioassay is technically valid and if potency results should be reported. It's typically required on each assay plate, and it should have specific numerical assay acceptance criteria. A staged approach for acceptance criteria setting is commonly accepted, and an acceptance criteria range is assigned using a statistically appropriate method to define the range and is based on a suitable sample size. Next slide, please. The implementation of an interassay control is recommended to be as early as possible, and is considered important as it allows the accumulation of data and assignment of relevant assay acceptance criteria to be established, which are, as previously mentioned, statistically appropriate based on the sample size. And this can be used to support bridging studies, for example, transitioning to a new batch of reference standard and used in comparability studies. Next slide, please. A review of the pros and cons upon implementing an ICE are described in this slide, and some of which include pros, include fewer out of trend and out of spec results, additional SST criteria to ensure assay performance and potency results, monitoring of intermediate precision, supportive data for reevaluation of reference standard, and enables the identification of a potential method or analyst bias, and enables the evaluation of the influence of replacement new reagent lots on assay performance. Some of the cons are that it is obviously an additional critical reagent, and alongside that is the responsibility for shelf life determination and assignment of storage conditions, additional documentation and costs. But more importantly, perhaps it does affect the plate layout and results in a lower sample throughput. Where some bioassay performers did not utilize controls, there are alternative control strategies to ensure adequate performance of their bioassays, and these included ED50 tracking and failure mode and effects analysis. Next slide, please. Once the initial stages of a bioassay development are complete, monitoring key assay performance parameters with a chronologically ordered trending chart, for example, Levi-Jennings control chart, can provide a valuable insight into assay performance. It can point out drift, indicate unexpected results due to instability of critical reagents, monitor the performance of the IC, and identify influences from formulation excipients and matrix effects. It is a useful tool to have in place for long-term monitoring of the bioassay performance, and is very useful if the bioassay is transferred to a secondary site with less expertise. Next slide, please.
Great, thank you, Samina. Next slide, please. So it's my pleasure to conclude this webinar. So as you heard from Jeff, the bioassay design is driven primarily by the mechanism of action of the drug. And this could be a complex mechanism of action involving 2 or more binding sites, including effector functions, or it could be a simple mechanism of action. The overarching design of the bioassay must include all these mechanisms of action. It could include multiple assays, each representing a single mechanism of action, or it could represent one assay, combining the mechanism of action into one assay. There's increasing challenges to support characterization. As Jeff explained, a lot of companies are now switching to the use of reporter assays and second messenger assays because they are more predictable in most cases, and also they can represent multiple mechanisms of action, and they're quite robust to implement. But we've also got to keep in mind that the bioassay being developed in the development organization has to move to a quality control environment. And so to achieve a robust assay, we need to think of several attributes of that assay. So the choice of method reagents, as was outlined, the choice of cell line, etc., the implementation of reagents, the design of the assay, the replication strategy, the elimination of the edge effect, all comes under the tactical design of the assay. And then we have to implement appropriate analytical tools. The 4-parameter is the most commonly used analytical approach, but it could include the 5-parameter. And then finally, the implementation of method control strategies, such as using system suitability, sample suitability, and as just explained, method trending, and also trending of multiple attributes of the assay over time could warn you that the assay is failing to perform for some reason. And we expect the assay design to evolve over the life cycle of the therapeutic. So most companies implement a binding assay, a lyso-based assay, for instance, to support phase 1 implementation. But then as the asset moves forward in the clinic to later phases development, more mature assays such as the cell-based assay are implemented. And that involves the level of control, allows you to implement the level of control on the assay and increase your product knowledge of the asset as well. So I can move to the next slide and talk about future perspectives. So there are multiple mechanisms of action. So a lot of us have dealt with the simple mechanisms of action, the interaction, the neutralization of a binding ligand. Companies are now implementing non-antibody approaches, but also antibodies with multiple mechanisms of action. Some antibodies have up to 3 or 4 mechanisms of action, all which must be represented or should be represented with a cell-based assay. There's also novel and emergent tools coming forward, such as the automation of platforms. This has driven a higher throughput, and in combination with reporter assays now leads to a more robust assay performance, which can be used in the next section for greater product understanding. If up to now, a lot of us have been limited by the number of samples we can run. The assays take multiple days to run. They have some failure rate, but with automation and reporter assays, we can implement much more robust cell-based assays, which lead to greater process and product understanding over time. Also, the sophisticated tools for data analysis, transforming data, as you heard before, and the meaning of that information. And some companies are reducing the reliance on cell-based assays for those that either have a single mechanism of action or for drugs which are in early phase of development. And you can do this because you can justify the use of those assays by having comparison or bridging studies. And so we're at the beginning of this journey, and each question leads to another question. So it actually looks like a very promising area, and the future will be very exciting for bioassay design. If I can go to the next slide. I'm not going to go through this slide, but just to acknowledge all the people who contributed to the paper and contributed to the webinar and other presentations which the BioPhorum group have made. Thank you.
Thanks very much, John.